Tonight, we're going to explore something that seems empty and far away, but is actually one of the most extreme realities on Earth: isolation. You may have seen these places on maps or in photos. Scientists talk about them when they study survival and limits. But here's what most people don't realize. When you truly understand how cut off these places are, when you grasp what makes life almost impossible there, it changes how you think about our planet. These are not just remote spots. They are environments so harsh and so alone that almost nothing can survive. And by the end of this journey, you're going to understand why these places exist, why life struggles to take hold, and why their silence tells us so much about Earth itself. Before we go any further, if you enjoy this kind of deep and simple exploration, a quick like or subscribe really helps the channel grow. It's a small thing for you, but it makes a big difference for me. Now, let's begin. On the surface of our planet, most people think of Earth as full of life, vibrant and active everywhere they look. Forests stretch across continents. Oceans teem with fish and mammals. Deserts are home to hardy plants and animals, and the air carries countless insects. Yet even in this vast living world, there are places where life struggles to survive, and some locations where nothing lives at all. These are the isolated corners of the Earth, areas shaped by extremes that push the limits of existence. Isolation, in its strictest sense, is not just a distance from human settlement, but an environment so harsh that organisms cannot sustain themselves. It is a combination of factors: temperature, water availability, pressure, chemical composition, radiation, and nutrient scarcity that create zones where the usual rules of life are suspended. Understanding these places is not only about mapping emptiness. It is about understanding the boundaries of life itself and the conditions under which organisms, including humans, cannot persist. The term isolation carries multiple meanings when applied to natural environments. Geographically, isolation may describe regions far from other land masses, where no species can easily reach or migrate. Ecologically, isolation may refer to environments so extreme that the survival of any organism is unlikely, regardless of how well it has adapted elsewhere. In some cases, these definitions overlap. Remote polar plateaus, high-altitude mountain peaks, hyper-arid deserts, deep ocean trenches, and volcanic wastelands all qualify in different ways. Each area demonstrates how Earth's physical processes can shape an environment into a place almost devoid of life. In these locations, the factors that usually support life, water, nutrients, warmth, shelter, are either absent or so inconsistent that they cannot sustain stable ecosystems. Isolation is thus not merely about being alone or distant. It is about the absence of the necessary conditions for life to establish and maintain itself. One of the first observations scientists make about these extreme places is that they are defined by absence. It is easier to describe what is missing than what exists. In the most isolated regions, there may be no visible plants, no animals, and sometimes not even signs of microbial activity on the surface. The landscape appears uniform and unbroken, often dominated by stone, ice, sand, or toxic sediments. In some cases, wind and erosion continually strip away any material that could support life, leaving surfaces barren and exposed. The absence is not only a visual phenomenon, it is measurable. Temperature records show constant extremes. Rainfall data indicate long-term aridity. Soil and water chemistry reveal toxicity, and radiation measurements demonstrate exposure levels beyond those tolerated by most organisms. In these environments, life is absent because the basic requirements for growth, reproduction, and maintenance cannot be consistently met. Isolation can also be seen in the way life fails to colonize these regions. Even where conditions are occasionally favorable for a brief period, organisms often cannot establish a permanent presence. Seeds carried by wind may land on sand or rock, only to dry and die. Microbial spores deposited by birds or air currents may find no moisture or nutrients to germinate. Animals attempting to migrate into these areas may be repelled by the harshness of the terrain or lack of shelter. Over time, natural selection favors those species that remain outside these extreme zones. Life, therefore, tends to cluster in areas that provide at least the minimal resources required to survive. Isolated places, by contrast, remain largely empty because the environmental pressures exceed the adaptive capacity of most organisms. Temperature is one of the primary forces creating isolation. In polar regions and high mountains, cold is relentless. Surface temperatures can drop to levels that freeze water, inhibit chemical reactions, and halt metabolic processes. In deserts, heat can soar to the point that protein structures in organisms are damaged. Water evaporates rapidly, and soil surfaces bake under the sun. Seasonal variations add complexity. Some areas experience long periods of extreme cold or heat, followed by brief windows of moderate conditions that are still insufficient for life to establish itself. Extremes do not only stress organisms, they prevent the formation of stable habitats. Rocks, ice, and shifting sands dominate, leaving few options for shelter, foraging, or reproduction. Water availability is another defining factor. Everywhere life exists, liquid water is essential. Isolated environments often lack a consistent supply. The interiors of Antarctica are not just cold, but hyper-arid. Some regions have received no measurable precipitation for centuries. Similarly, the Atacama Desert in South America is one of the driest places on Earth, with some areas recording less than a millimeter of rainfall per year. Salt flats, brine lakes, and volcanic slopes may contain water, but its chemical composition, high acidity, salinity, or toxicity renders it unusable for most life forms. Without water that can be absorbed and metabolized, organisms cannot survive. Even microbes, which can endure in extreme conditions elsewhere, are constrained by the complete absence or chemical hostility of water in these zones. Chemical composition and soil properties also determine isolation. Many barren areas have soils or sediments that are inhospitable due to acidity, alkalinity, salinity, or heavy metal concentrations. In volcanic regions, lava flows and ash deposits create sterile surfaces that take decades or centuries to develop even minimal microbial communities. In deserts, soils are often so nutrient-poor that plants cannot grow, and the limited biomass cannot support animals. Minerals present may bind available water or release toxic ions, further reducing habitability. In effect, the chemical environment acts as a barrier, preventing colonization even when temperature and moisture might occasionally allow it. Life is therefore not simply absent by chance. It is actively constrained by chemical conditions that exceed tolerance thresholds. Wind and sunlight add another layer to the challenge of survival. In open, exposed regions, strong winds strip away loose soil, sand, or organic material, preventing the accumulation of a surface capable of supporting plants. Solar radiation, particularly ultraviolet light at high altitudes or in polar regions, can damage DNA and cellular structures in organisms. The combination of constant erosion, abrasive winds, and radiation exposure creates a surface environment that is continuously hostile. Even where small pockets of shelter exist, Their isolation can prevent species from establishing a broader population. These forces are subtle but powerful in shaping the patterns of life, or the absence of it, across isolated landscapes. Extreme pressure and depth define isolation in other ways. In the deep oceans, sunlight cannot penetrate, and pressure increases by hundreds of times that at the surface. Nutrient availability is minimal, and organic material must drift down from the upper layers to sustain even the smallest organisms. Life does exist in these depths, but it is highly specialized and sparse. Large animals struggle to survive, and ecosystems are scattered. In this sense, isolation is not only horizontal across distance, but vertical, along gradients of pressure and energy that make large areas inhospitable. The deepest trenches of the ocean floor are therefore effectively lifeless zones for most forms of life, despite the potential for microbial extremophiles to exist. Geography and distance play their part as well. Islands and remote rocks far from continents can be biologically empty. Seeds, spores, and small animals rarely reach these places, and when they do, they face harsh environmental conditions. Ocean currents, wind patterns, and climatic isolation prevent the establishment of permanent populations. Even when these places are occasionally visited by birds or drifting debris, life cannot anchor itself. These isolated spots are defined not only by what they lack in resources, but also by what they lack in connectivity. Dispersal limitations compound environmental stress, creating places that remain almost entirely empty over time. Understanding these isolated places is critical for science. They serve as natural laboratories for studying the limits of life. By examining why life cannot survive, scientists learn about the conditions necessary for ecosystems, the ways organisms adapt to extreme stress, and the thresholds beyond which survival becomes impossible. Isolation highlights the delicate balance of factors that sustain life. It also provides insight into planetary processes, from glaciation to volcanism, that shape the landscape and define habitability. By observing these barren zones, researchers gain a deeper understanding of life's resilience and fragility, and the conditions under which Earth's biosphere operates at its outermost limits. These regions, empty as they may seem, are rich with knowledge about the dynamics of survival and the boundaries of existence itself. The heart of Antarctica stretches far beyond the familiar images of icy coastlines and research stations. Inland, the landscape becomes a vast expanse of ice, rock, and snow, stretching as far as the eye can see. Mountains rise in jagged lines, but most of the continent is covered by an immense ice sheet that holds nearly 90% of the world's fresh water. The interior is a place where sunlight is weak for months, and then absent entirely for long winters. Temperatures drop to levels that are almost impossible to survive. Winds sweep across the ice with a force that strips moisture from the land and chills any exposed surface. In these conditions, life is almost entirely absent. Only in rare pockets can microbes endure, and even these are few compared to regions just a few hundred kilometers closer to the coast. The interior is a true dead zone, a land defined by absence and extremes. The geography of Antarctica shapes its isolation. The continent is surrounded by ocean, but largely insulated from it by strong winds and the vast ice sheet. The ice is thick and heavy, sometimes stretching several kilometers deep, creating a surface that is smooth in some areas and broken by ridges, crevasses, and exposed rock in others. Mountains appear as isolated peaks, called nunataks, emerging from the ice like islands in a frozen sea. These peaks often remain snow-free in places where winds scour the surface, but even they are inhospitable. The ice sheet moves slowly, carrying snow and debris, shaping valleys and slopes, and covering areas that might otherwise support life. The movement and thickness of the ice form a barrier that prevents most organisms from reaching the interior, and even if they do, survival is nearly impossible. Sub-zero temperatures define the Antarctic interior. In the coldest months, thermometers can register below -80 degrees Celsius, and even in summer, conditions remain far below freezing. At these temperatures, water is locked in ice, unavailable for life to use. Chemical reactions in cells slow or stop completely. Proteins and enzymes which enable life's processes cannot function in the prolonged cold. Microbial life is sometimes found in thin films of moisture around rocks or within ice, but these organisms grow slowly and sparsely. The extreme cold prevents plants and animals from establishing populations, and the interior becomes a place where the usual patterns of life do not occur. Even microscopic life struggles to survive for more than brief periods or in very small numbers. Wind is another force that isolates Antarctica's interior. Katabatic winds, which flow down from the high interior, can reach speeds over 200 kilometers per hour. These winds carry ice crystals and snow, grinding surfaces and stripping away any small deposits of soil or organic matter. The air is extremely dry, removing moisture from any exposed organisms and preventing the formation of liquid water at the surface. Wind and cold together create an environment where only the most extreme microbes, capable of enduring desiccation and freezing, can exist. Even birds or insects, which can occasionally travel long distances, find these regions uninhabitable. The winds erase traces of life before they can establish themselves, maintaining the barrenness of the interior. The ice deserts of Antarctica are areas where snow is minimal and life is almost entirely absent. In places such as the McMurdo Dry Valleys, snow and ice are swept away or sublimated by wind and low humidity. These valleys have been compared to Mars in their barrenness, with bare rock and gravel stretching over large areas. Despite occasional liquid water in small streams or melted ponds during brief summers, the chemical and thermal conditions remain so extreme that only microbial life survives. Experiments have shown that microbial communities exist in thin layers of soil or within rocks, but even these are limited in diversity and abundance. The absence of plants, insects, and larger animals highlights the extremity of these deserts, where the combination of cold, dryness, and wind creates a nearly lifeless land. Scientific expeditions have long encountered these dead zones with surprise and awe. Early explorers who penetrated inland often expected to find some forms of life, yet they observed vast white expanses with no visible flora or fauna. Notes from these journeys describe surfaces that appear untouched for centuries, without tracks or signs of animals. In some areas, exposed rocks show no lichen or moss. Scientists, visiting in more modern times, have confirmed these observations using satellite imagery, soil sampling, and on-site microbial studies. Even in the most extreme conditions, life exists only in hidden niches, often beneath stones or within thin films of brine. The contrast between coastal regions, which may support seals, penguins, and microbial mats, and the interior, which hosts virtually nothing, illustrates the starkness of isolation in polar extremes. Ice sheets themselves play a central role in preventing colonization. The thickness of the ice, sometimes several kilometers, creates a continuous barrier over the land, covering rock surfaces, burying valleys, and preventing the accumulation of soil. Snow and ice movement can crush or sweep away any organisms that attempt to colonize the surface. In addition, the reflectivity of ice, known as albedo, keeps temperatures low and limits the amount of heat absorbed from the sun. The ice acts as a physical, thermal, and hydrological barrier, creating a dynamic but inhospitable environment. Only at the edges, where the ice is thinner or retreats during summer, can life occasionally take hold, and even then, it is fragile and temporary. Microbial life in these zones demonstrates the limits of survival. Bacteria and archaea can survive in thin layers of moisture trapped within rocks or ice, forming tiny colonies that metabolize slowly. Some use chemical reactions with minerals rather than sunlight to generate energy. These organisms grow at temperatures near freezing, resist drying out, and endure high ultraviolet radiation. Despite these adaptations, their populations are sparse and localized, incapable of forming extensive ecosystems. There are no plants, no animals, and very few predators or prey. The interior of Antarctica thus remains a laboratory for understanding how life can persist in extreme conditions and where its boundaries lie. Even with advances in technology, human presence does not change the fundamental isolation of these areas. Scientific stations near the coast provide controlled environments where life can be studied and maintained, but they do not significantly alter the lifelessness of the interior. Drones, snow vehicles, and remote sensors can traverse the ice, but they encounter the same empty landscapes that explorers faced a century ago. Observations continue to reinforce the idea that some places are naturally resistant to colonization. The interior ice deserts are defined not only by their harshness, but by the inability of organisms, regardless of adaptations, to establish and maintain populations over time. In summary, Antarctica's interior exemplifies the extreme isolation that occurs on Earth. The combination of geography, sub-zero temperatures, katabatic winds, and ice sheet coverage creates an environment almost entirely devoid of life. Microbial communities survive in hidden niches, but no larger organisms can persist. Historical accounts and modern, scientific observations both confirm that this vast interior is a true dead zone, a place where the forces of nature outweigh the capacity of life to endure. The continent's interior illustrates the boundaries of habitability, showing how Earth's most isolated regions remain beyond the reach of most living things, offering insight into the limits of life and the resilience of the few organisms that manage to endure. Moving from the frozen silence of Antarctica, the Earth's most isolated regions also exist in deserts, where heat and dryness create environments hostile to life. The Atacama Desert in northern Chile stretches along the Pacific coast, a land that has earned the title of the driest place on Earth. Rainfall is almost non-existent, with some areas receiving less than a millimeter per year. In other parts, decades may pass without measurable precipitation. The surface is cracked, dusty, and covered with salt deposits left behind as ancient waters evaporated long ago. Winds sweep across the terrain, lifting dust and eroding small rocks, leaving behind flat plains and jagged ridges of salt. Life here is extremely limited, constrained to the most hidden and resilient microbial communities. The Atacama offers a striking example of how dryness alone can limit life, producing vast areas where macroscopic organisms, such as plants and animals, cannot survive. The Danakil Desert in Ethiopia presents a different set of challenges. Its landscape is dominated by salt flats, volcanic craters, and sulfuric springs. Temperatures frequently exceed 40 degrees Celsius, and the air is thick with chemical vapors. The soil is rich in minerals, including salt and sulfur compounds, but lacks the water necessary to sustain conventional life. The extreme heat and chemical toxicity of the terrain make it nearly impossible for plants or animals to persist. Only specially adapted microbes can tolerate these conditions, using chemical reactions with the minerals in the soil as a source of energy. Even for microbes, survival is limited to specific niches where moisture or chemical gradients allow metabolic activity to continue. Large-scale life is absent, leaving the desert almost entirely barren to the human eye. Across the African continent, parts of the Sahara Desert offer another perspective on hyper-arid isolation. Vast sand seas stretch endlessly, with dunes shifting under the influence of dry winds. Sunlight is intense, and temperatures during the day soar well above 40 degrees Celsius, dropping drastically at night. Water is scarce, appearing only in ephemeral oases or in occasional rainfall that evaporates quickly. In some regions, sand and rock surfaces are covered with crusts of minerals and salts that inhibit the growth of any plants. Life is concentrated in isolated patches where soil can retain moisture, or where underground aquifers feed vegetation. The contrast between these small pockets and the surrounding barren expanses highlights the extreme limitations placed on organisms by heat and lack of water. The absence of macroscopic life in hyper-arid deserts is directly tied to water scarcity. Liquid water is essential for metabolism, growth, and reproduction in all known forms of life. In the Atacama and similar deserts, the combination of high temperatures and low humidity means that water evaporates almost immediately when it appears. Any rainfall is quickly absorbed by the soil or lost to the atmosphere. Plants cannot root in dry soil, and animals cannot rely on intermittent moisture for survival. Microbial life, by contrast, can endure in microscopic niches, often within rocks, salt crusts, or thin films of moisture formed from fog or dew. These microbes may remain dormant for years, awaiting conditions that allow limited metabolic activity, but they never form visible colonies that could support larger food chains. Soil chemistry in these deserts further restricts life. Salt deposits and mineral-rich sediments dominate many areas, creating conditions that are toxic to most plants and animals. High concentrations of salts disrupt the ability of cells to retain water, leading to dehydration and death. Sulfur, arsenic, and other mineral compounds present in volcanic or evaporated soils can inhibit biological processes, leaving the terrain uninhabitable for conventional organisms. Even where water is briefly available, chemical toxicity prevents life from establishing itself. Microbes, however, have developed adaptations that allow them to survive in these hostile soils. Halophilic organisms, for instance, thrive in high salt conditions by maintaining internal balances of ions that prevent dehydration. Acidophils can metabolize in highly acidic soils that would destroy other cells. Despite these adaptations, microbial life is sparse and isolated, and the landscape remains largely lifeless in its appearance. Comparison of microbial life and macroscopic organisms illustrates the extremes of isolation. In the Atacama, or Danakil, microbes can colonize surfaces or exist within mineral layers, but they do not form large, visible communities. Mosses, lichens, or small plants may appear in rare sheltered locations, but they cannot reproduce widely. Insects, reptiles, and mammals are almost entirely absent. The energy available in these deserts, combined with water and nutrient limitations, cannot sustain larger organisms that require more substantial resources. Microbial extremophiles survive quietly and slowly, while macroscopic life fails to establish a presence. This contrast underscores how environmental factors determine which forms of life can persist and which are excluded entirely. Hyper-arid deserts are shaped not only by current climate, but by geological and atmospheric history. Many areas were once wetter, and their present dryness results from long-term changes in rainfall patterns, wind circulation, and tectonic activity. Salt flats, such as those in the Atacama, are remnants of evaporated lakes, leaving behind thick layers of minerals. Volcanic activity in the Danakil region has deposited lava and ash that chemically alter the soil and create new barriers to life. In the Sahara, sand seas formed from wind-driven erosion, and rock surfaces were exposed over thousands of years. These geological factors combine with climate extremes to produce the isolation that characterizes these deserts today. Even small fluctuations in conditions rarely allow life to expand. Occasional fog in the Atacama provides moisture for thin microbial films. Yet this is temporary. Rainfall in the Danakil is insufficient to support plants beyond short-lived algae mats. In the Sahara, underground aquifers can sustain oases, but the surrounding sand plains remain empty. The isolation of these deserts is therefore a combination of multiple factors: water scarcity, high heat, chemical challenges, wind erosion, and unstable surfaces. Each element reinforces the others, creating areas where life cannot gain a foothold beyond microscopic organisms in specific, sheltered locations. Scientific study of these deserts has revealed how life can exist at the edge of possibility. Researchers collect soil samples, analyze chemical compositions, and examine microbial communities using microscopy and genetic sequencing. They have discovered organisms capable of surviving with almost no water, metabolizing minerals, and enduring extreme temperatures. These studies provide insight into life's resilience and the minimum conditions required for existence. Yet the broader landscape remains strikingly empty, emphasizing the contrast between isolated microbial survival and the absence of macroscopic ecosystems. The hyper-arid deserts of the world stand as reminders that life is not omnipresent. Even on a planet abundant with organisms, areas exist where survival is impossible for all but the most specialized microbes. These regions teach scientists about adaptation, stress tolerance, and the boundaries of biological activity. They also illustrate the limits of colonization and ecosystem formation. Where water is absent, heat is extreme, and soils are chemically hostile, the desert maintains its isolation, a land defined not by what is present, but by what can't endure. Life survives at the margins, leaving the majority of the terrain lifeless, stark, and profoundly isolated. Beneath the waves of the world's oceans lie regions that few humans will ever see, depths where light cannot reach and pressure rises to crushing levels. The Mariana Trench, the deepest known part of the ocean, plunges nearly 11 kilometers below the surface. It is a place of constant darkness, near freezing temperatures, and enormous pressure, over 1,000 times what exists at sea level. Life in these depths is sparse, and the conditions are so extreme that most familiar animals cannot survive. Large fish, mammals, and plants are absent. The trench is effectively a dead zone for macroscopic organisms, yet it is inhabited by small, highly specialized creatures capable of enduring the harsh environment. These deep ocean trenches illustrate another form of isolation, one defined not by dryness or cold, but by depth, pressure, and the scarcity of nutrients. The pressure at the bottom of the trenches is one of the primary barriers to life. Water at these depths presses down with a force that can crush most organisms. Cells not adapted to these conditions cannot maintain their structure, and tissues collapse under the immense weight. Even skeletal frameworks struggle to withstand the constant compression. Life persists only when organisms possess special adaptations. Some deep-sea fish have flexible, gelatinous bodies that resist compression. Others produce cellular chemicals that stabilize proteins and membranes against the high pressure. Invertebrates, such as certain worms and crustaceans, often have reduced skeletons or exoskeletons capable of tolerating the forces that dominate the Hadal zone. Without these adaptations, survival is impossible, and the trench remains off-limits to the majority of ocean life. Darkness is another defining feature of the Hadal environment. Sunlight fades within the first few hundred meters of ocean, leaving the trench in complete black. Photosynthesis, the primary source of energy for life in shallower waters, cannot occur here. Energy must arrive in other forms, such as organic material sinking from the upper layers, or chemosynthesis powered by chemicals from the Earth's crust. The absence of light also affects behavior and physiology. Eyes may be reduced or absent. Bioluminescence becomes common, and sensory systems other than sight are enhanced. Life that cannot adapt to darkness fails to survive, contributing to the emptiness of the deep. The trench is a place where visibility and vision are irrelevant, and survival depends on entirely different strategies from those of the surface. Nutrient availability in deep ocean trenches is extremely low. Most of the food comes from detritus drifting down from shallower waters, often called marine snow. This consists of dead plankton, waste products, and small organic particles. At great depths, much of it is consumed before it can reach the trench floor, leaving only minimal sustenance. In some areas, hydrothermal vents provide localized sources of energy, supporting microbial mats and small animal communities. However, these vents are rare, and their influence does not extend far. As a result, life in trenches is sparse, scattered, and slow-growing. Animals cannot accumulate enough energy to grow large, reproduce quickly, or sustain complex food webs. The isolation created by nutrient scarcity limits biodiversity and keeps the environment largely lifeless by comparison to surface waters. Large life struggles to survive under these combined conditions. Fish cannot grow to the sizes seen in coastal waters because energy is insufficient and pressure imposes structural constraints. Mammals cannot exist at these depths because they require more oxygen, food, and movement space than the trench allows. Even some invertebrates that survive shallow waters are absent, replaced by species specialized for high pressure and low energy. Growth rates are slow, reproduction is infrequent, and populations are small. The trench is effectively a barrier to conventional life, and only organisms with extreme adaptations can persist. The result is a community of organisms unlike any found elsewhere, sparse, isolated, and highly specialized. Extremophile species demonstrate life's capacity to endure. Bacteria and archaea dominate the microbial populations, using chemosynthesis or metabolizing detritus to sustain themselves. Small crustaceans, amphipods, and polychaete worms feed on the limited organic material that reaches the trench floor. Certain fish, such as the snailfish, possess soft, flexible bodies and enhanced chemical sensing that allow them to detect food and mates in darkness. These organisms live slowly, reproduce cautiously, and occupy microhabitats where competition is minimal. Extremophiles in the Hadal zone illustrate the ability of life to adapt to extreme pressure, darkness, and scarcity, but their existence does not counterbalance the overall lifelessness of the environment. Large ecosystems cannot form, and the trench remains largely empty to the human eye. Isolation in the Hadal zone affects biodiversity in a pronounced way. Many species are found only in a single trench, or even in a narrow depth range within that trench. Dispersal is limited because the extreme conditions prevent movement across larger distances. Surface currents, which transport plankton and larvae in shallower waters, do not extend into the deepest layers. Geological barriers and water density gradients further limit colonization. As a result, life in the trenches is fragmented, with small populations separated by vast, inhospitable distances. Evolution proceeds slowly, and ecosystems remain simple, consisting of microbial mats, small invertebrates, and highly specialized fish. The biodiversity of these regions is low, and isolation ensures that life does not expand beyond these narrow confines. Temperature in the Hadal zone adds another layer of challenge. Near freezing waters prevail, and even thermal vents provide only small localized warmth. Enzyme activity, metabolism, and movement are all constrained by the cold. Organisms that cannot tolerate the temperature cannot survive, and only cold-adapted species persist. Combined with pressure, darkness, and food scarcity, the cold ensures that large familiar forms of life cannot exist. Microbes dominate because they can grow slowly, remain dormant for long periods, and survive on the minimal energy available. The trench becomes a world defined by absence, punctuated only by organisms highly adapted to extreme conditions. The trench floor is often soft, composed of fine sediments accumulated over centuries. These sediments may contain chemical compounds, remnants of organic material, or minerals leached from the surrounding rock. Microbes interact with these sediments, extracting energy and sustaining minimal communities. Small invertebrates burrow or move across the sediment, feeding on microbial films or detritus. The surface appears flat and uniform, but beneath this apparent stillness, slow biological processes continue. Nevertheless, these processes do not create complex ecosystems. Life is small, scattered, and invisible to casual observation. The trench floor is a quiet testament to isolation, a world where survival occurs in slow, subtle ways, rather than through the abundance and diversity seen elsewhere. Even in trenches that receive occasional large inputs of nutrients, life remains constrained. Dead animals or drifting material may attract small swarms of amphipods or worms, but these events are rare and temporary. The scarcity of food and extreme conditions prevent populations from growing, and organisms retreat to safer niches afterward. Isolation is maintained because the environment cannot sustain continuous, abundant life. Trenches demonstrate how extreme pressure, darkness, and low nutrients combine to create regions where ecosystems cannot thrive. Large animals fail to exist, complex food webs cannot develop, and life is reduced to specialized small-scale processes. Scientific exploration of these depths has revealed much about the limits of life. Robotic submersibles, deep-sea landers, and remotely operated vehicles have photographed the trench floor, collected specimens, and measured physical conditions. Observations confirm that while life exists, it is sparse, slow-growing, and specialized. Experiments show that microbes can survive extreme pressure, and small animals can endure long periods without food. These studies offer insight into how organisms adapt to conditions that would be lethal elsewhere. The trenches serve as natural laboratories for understanding pressure tolerance, energy limitation, and the biology of isolation, revealing how the boundaries of life can be stretched but not removed entirely. In conclusion, deep ocean trenches like the Mariana Trench represent one of Earth's most isolated environments. Darkness, extreme pressure, low nutrient availability, and cold temperatures combine to create areas largely devoid of macroscopic life. Extremophiles endure in small niches, and biodiversity is limited to a few specialized species. Isolation is not only physical but ecological, preventing colonization and sustaining sparsely distributed populations. These trenches show that even in a water-rich world, certain environments remain nearly empty, defined by the extreme challenges that limit survival and prevent life from spreading beyond its narrow confines. Far above sea level, where the air grows thin and the landscape stretches toward the sky, another form of isolation emerges. The high mountains of the world, ranges like the Andes, the Himalayas, and the Tibetan Plateau, present conditions that limit life in ways both subtle and extreme. As altitude increases, the atmosphere thins, reducing oxygen levels for respiration. Solar radiation intensifies because there is less air to filter ultraviolet rays. Temperatures fluctuate dramatically between day and night, and surfaces are often rocky, unstable, and exposed to wind. Above certain heights, these factors combine to make the environment almost uninhabitable. Flora becomes sparse, animals are few, and human presence is rare. Life in these high-altitude zones must adapt to extremes, and in many locations, the combination of conditions leaves the terrain almost empty. The Andes, stretching along the western edge of South America, offer a dramatic example. At elevations above 5,000 meters, the air is thin, with roughly half the oxygen found at sea level. Plants cannot rely on steady growth, as frost can strike even in summer, and sunlight is intense. Animals, too, must adjust. Few species can tolerate the low oxygen and cold simultaneously. Specialized birds, such as the Andean condor, exploit thermals and travel widely to find food, but most mammals are limited to lower valleys. In isolated plateaus, grasses and mosses cling to cracks in the rock, but trees cannot survive. Water is often locked in glaciers or snowfields, leaving the soil dry. The Andes illustrate how altitude creates both physical and ecological barriers that prevent most life from expanding. The Himalayas are even more extreme. Peaks rise above 8,000 meters, entering what mountaineers call the death zone, where human survival is measured in hours without oxygen support. Temperatures plunge far below freezing, and winds can reach hurricane speeds. At these heights, no permanent human settlement exists, and most plants and animals are absent. Some microbes and extremophiles survive in small pockets of soil or within ice, but these populations are minimal. Large birds, insects, or mammals cannot sustain themselves on the sparse food supply. Even temporary colonizers are few, retreating to lower altitudes to survive. The Himalayas demonstrate that life is constrained not only by temperature and oxygen, but also by the thinness of the atmosphere and exposure to radiation. The Tibetan Plateau, a vast highland averaging more than 4,000 meters above sea level, combines isolation with harsh climatic conditions. The plateau experiences intense sunlight, cold winters, and low rainfall. Permafrost underlies much of the soil, limiting plant roots. Only hardy grasses, sedges, and small shrubs survive in these regions, forming sparse communities. Animals, such as yaks and wild goats, exist only in lower or sheltered areas where grazing is possible. Water is unevenly distributed, with rivers fed by melting glaciers supporting life in otherwise barren areas. The plateau's isolation results from both its altitude and its geological history, which has created a high, flat expanse, difficult for species to traverse or colonize from surrounding regions. thin atmosphere at high elevations is a constant challenge. Oxygen levels drop steadily as altitude rises, and low oxygen impairs respiration for animals and limits photosynthesis in plants. Some species have developed adaptations, such as increased lung capacity, higher hemoglobin levels, or specialized enzymes that allow metabolic activity at low oxygen. Plants at these heights grow slowly, reproduce irregularly, and are usually small. Without these adaptations, life cannot survive. The lack of oxygen creates a natural barrier, ensuring that many species remain below certain elevations. Combined with temperature extremes and limited water, the thin air maintains isolation across high mountain regions. Radiation intensifies with altitude, adding another layer of isolation. Ultraviolet rays are less filtered by the thinner atmosphere, and surfaces exposed to sunlight receive higher energy levels than at lower elevations. This radiation can damage DNA and cellular structures in plants, animals, and microbes. Organisms must develop protective pigments, thick outer layers, or behavioral strategies, such as burrowing or moving during certain hours. Even with these adaptations, life remains limited. On snow-covered slopes or rocky ridges, little vegetation exists, and animals are transient. Radiation, combined with temperature swings and low oxygen, reinforces the inhospitable character of high-altitude zones. Temperature swings in these regions are dramatic. Days may be relatively warm under direct sunlight, but nights can plunge far below freezing. Frost can occur during summer, and ice persists year-round in many areas. Soil can freeze and thaw repeatedly, disrupting plant roots and microbial communities. Wind enhances heat loss and can scour surfaces, removing soil or small vegetation. Organisms that survive at high altitudes often have slow growth rates, long life cycles, and small populations. Animals may migrate or hibernate to cope with seasonal extremes. The variability of temperature adds to the isolation, making high mountains harsh and difficult places to sustain life. Flora and fauna become increasingly scarce with elevation. Trees are generally absent above certain heights, replaced by small shrubs, mosses, lichens, and hardy grasses. Insects are rare, and mammals are mostly limited to lower slopes. Birds may nest in exposed cliffs, but often travel great distances to feed. Microbes persist in cracks, soil, or snow patches, forming the foundation of life in these high places. The scarcity of larger organisms illustrates how multiple environmental factors, low oxygen, radiation, cold, and wind, combine to prevent colonization. Life at these heights exists in a fragile balance, confined to microhabitats and specialized niches. Geological factors contribute to isolation as well. Mountain ranges create steep slopes, deep valleys, and fragmented terrain that is difficult for species to cross. Rockfalls, avalanches, and shifting glaciers remove soil and destroy established plant life. The fragmentation prevents populations from spreading and limits gene flow, further isolating surviving species. Plate tectonics that created these mountains also uplifted the land, exposing organisms to new extremes rapidly in geological terms. Many species cannot adapt quickly enough to changing altitudes, leaving only the most flexible or slow-growing microbes. The combination of steep geography, unstable surfaces, and harsh climate ensures that high mountains remain sparsely populated by life. Even human presence is rare and temporary at high altitudes. Explorers, mountaineers, and scientists visit extreme peaks. But settlements are limited to lower valleys. Conditions prevent agriculture, water supply is difficult, and oxygen is low. Human activity does not alter the isolation of the high-altitude ecosystem. Microbial extremophiles may continue to exist, and a few hardy plants cling to rock surfaces, but the overall landscape remains barren. The mountains illustrate how elevation alone can enforce isolation, creating regions where life exists only at the edge of possibility. High-altitude zones demonstrate that isolation is multidimensional. It is not only distance from other habitats, but also the combination of environmental stressors that makes life impossible for most species. Temperature, oxygen, radiation, wind, and unstable geology work together to limit survival. Life persists only in small niches, and larger organisms are confined to lower areas. High mountains show that even in areas of Earth with abundant light and open space, the environment can be too extreme for colonization. They are examples of isolation created by natural forces, revealing the boundaries within which life can exist. In conclusion, the Andes, Himalayas, and Tibetan Plateau represent some of the most isolated places on Earth by altitude. The thin atmosphere, intense radiation, extreme temperature swings, and rugged terrain limit life to microbes and a few specialized plants and animals. Isolation is reinforced by geology, slope, and soil instability. These regions highlight how extreme conditions can shape ecosystems, constrain biodiversity, and create vast areas where life struggles or fails to persist. High-altitude zones remind us that isolation on Earth is not only a matter of distance or remoteness, but also of environmental limits that define the range of living organisms. Volcanic regions on Earth form another type of isolated environment. These areas are defined by heat, chemical toxicity, and barren surfaces. Islands like Deception Island in Antarctica, the slopes of Mount Rainier in North America, and large lava fields in Hawaii or Iceland demonstrate how volcanic activity creates zones where life struggles to survive. Fresh lava and ash cover the land, destroying plants, soil, and small animals. Chemical compounds released during eruptions, sulfur, carbon dioxide, and other gases, alter the air, water, and soil, creating conditions inhospitable to most forms of life. These volcanic wastelands are rarely occupied by humans, except for short-term scientific studies, and even microbes must overcome extreme challenges to persist. The isolation of these areas is defined not by distance alone, but by the natural forces that shape the land and prevent colonization. Deception Island is an example of volcanic isolation in a polar setting, formed by a caldera that collapsed and filled with seawater. The island's interior is a mix of ash, frozen ground, and hot springs. Recent eruptions have coated large sections of the land with fresh volcanic deposits, sterilizing surfaces and removing the limited vegetation that had taken root. Temperatures near hot springs may allow some microbial activity, but most of the island remains lifeless. Chemical conditions in the soil and water further restrict life. High concentrations of salts, acids, and metals combine with extreme cold to prevent most plants and animals from surviving. Only a few specially adapted microbes can colonize the area, and even these populations remain small and localized. Observers often note the stark contrast between these volcanic zones and nearby areas that have recovered enough to support mosses or lichens. On the slopes of Mount Rainier, volcanic soil and repeated eruptions create barren patches. Lava flows and pyroclastic deposits cover large areas, stripping away existing vegetation. Soil is thin, rocky, and chemically altered, containing minerals and elements toxic to most plants. In some areas, streams of acidic water leach nutrients from the soil, further preventing life from establishing. Plants that colonize must tolerate heat, lack of moisture, and chemical stress. Early colonizers are typically microbial mats, bacteria, and lichens that slowly accumulate organic matter. Over decades or centuries, these communities may allow more complex plants to take root. The pace of life recovery is slow, and large animals are absent except for those able to move from nearby undisturbed areas. These volcanic slopes illustrate how environmental stress and chemical toxicity combine to create isolation, limiting biodiversity and slowing ecological succession. Lava fields, such as those in Hawaii and Iceland, are other examples of extreme isolation. Fresh lava creates solid rock surfaces with minimal cracks, no soil, and almost no moisture. Sunlight may heat these rocks to extreme temperatures during the day, while nights become cold. Wind and rain may slowly erode surfaces, allowing tiny pockets where microbial life can begin. These microbes can survive on trace nutrients and form biofilms on rock surfaces. Fungi and lichen may colonize later, producing small amounts of organic material that can support insects or small plants. The recovery of life is a slow, stepwise process, and until these initial colonizers establish themselves, the lava field remains nearly lifeless. The scale of isolation is magnified by the inhospitable surface, exposure, and lack of nutrients. Chemical toxicity in volcanic areas is a major barrier to life. Gases released during eruptions can linger in soils and water. Sulfur dioxide, carbon dioxide, and hydrogen sulfide create acidic conditions that prevent most plants and animals from surviving. Minerals present in lava and ash may bind water or release toxic ions. Even when water is present from rain or melting snow, its chemical composition may be unsuitable for growth. Microbes that survive these conditions often metabolize minerals directly, extracting energy from chemical reactions rather than sunlight. This chemical isolation means that even regions with some moisture or warmth remain largely uninhabitable for conventional life. Early colonization in volcanic wastelands typically begins with microbes. Bacteria and archaea can settle in tiny cracks in rocks, soil, or ash. They metabolize trace minerals and slowly produce organic matter. Lichens and mosses may follow, forming small patches that increase soil stability and fertility. These pioneer organisms establish the foundation for larger communities, but the process is slow. In some areas, decades may pass before higher plants take root, and centuries before more complex ecosystems develop. Large animals generally do not inhabit these zones until food and shelter become available, which may take hundreds of years. The pace of ecological recovery demonstrates how isolation in volcanic regions is not absolute, but gradual, mediated by the harshness of the environment. Human observations of volcanic wastelands provide insight into the scale and nature of isolation. Early explorers and scientists noted barren landscapes after eruptions, with no signs of life and surfaces that appeared permanently sterilized. Modern studies use soil sampling, remote sensing, and microbial analysis to track colonization and recovery. Experiments show that microbes can survive heat, chemical stress, and desiccation, but the spread of life is limited by the harsh conditions. Observers also note that volcanic eruptions can reset ecosystems, temporarily expanding areas of isolation. Life may return slowly, but the pace is constrained by environmental extremes. These studies highlight how isolation is dynamic and closely tied to geological processes. The heat of volcanic surfaces adds a further challenge. Newly erupted lava can reach temperatures over 1,000 degrees Celsius. Rocks remain hot for days, weeks, or months, depending on size and composition. These conditions prevent seeds from germinating and animals from foraging or nesting. even microbes may be excluded until surfaces cool sufficiently. Heat, combined with chemical toxicity and lack of soil, reinforces the lifeless character of these landscapes. Life is constrained to microhabitats, shaded areas, or cooled surfaces, where conditions are slightly less extreme. The combination of physical and chemical stress maintains isolation over time. Volcanic ash deposits also affect life. Ash can smother existing vegetation, cover nutrient-rich soil, and create unstable surfaces. Layers of ash compact over time, forming crusts that are difficult for plants to penetrate. Water infiltrates slowly, and mineral composition may inhibit germination. Microbial colonization is possible, but limited to surface layers or cracks. The physical structure of ash contributes to the isolation by preventing soil development and restricting the growth of larger plants. Animals are excluded unless they can migrate from surrounding undisturbed regions. Despite these challenges, life eventually returns in subtle ways. Microbial extremophiles thrive in cracks, under rocks, or in small pools of water. Lichens and mosses may colonize, producing organic matter that gradually improves soil fertility. Insects, small invertebrates, and eventually plants can appear as conditions allow. This slow recovery demonstrates the resilience of life, but also emphasizes the magnitude of isolation created by volcanic environments. For long periods, these regions remain barren, and the ecological footprint of life is minimal compared to surrounding areas. The isolation is therefore both immediate after eruptions and sustained by harsh conditions over time. Scientific experiments in these areas often involve introducing microbes to sterilize lava or testing soil chemistry to understand the limits of colonization. Researchers study how chemical stress, heat, and lack of nutrients interact to limit growth. They observe microbial succession, the first signs of moss or lichen establishment, and the gradual increase in biodiversity. Such experiments confirm that volcanic wastelands are extreme environments where isolation is defined by multiple overlapping factors. The landscape may look empty, but it serves as a laboratory for understanding how life persists under the harshest conditions. In conclusion, volcanic wastelands and toxic environments, such as Deception Island, Mount Rainier slopes, and lava fields, are among Earth's most isolated regions. Heat, chemical toxicity, lack of soil, and unstable surfaces prevent most plants and animals from surviving. Microbes colonize first, initiating slow ecological succession, while larger life remains absent until conditions improve. Human observations and experiments have confirmed that these environments are defined by extreme stress, limited resources, and persistent isolation. Volcanic zones reveal how Earth's natural processes can create lifeless areas, showing the boundaries of colonization and the slow pace at which life can re-establish itself in hostile terrain. Across continents, there are landscapes where water exists but is so salty, alkaline, or chemically unusual that life struggles to survive. Salt flats, brine lakes, and chemical pools form some of the most isolated and lifeless environments on Earth. The Salar de Uyuni in Bolivia, the vast expanse of white salt stretching for over 10,000 square kilometers, is an extreme example. The surface is flat and reflective, composed of thick layers of crystallized salt. Rain is minimal, and when it does fall, it forms shallow pools that evaporate quickly, leaving behind more salt. No plants grow here, and animals are almost entirely absent. The only life consists of microbes that have adapted to the extreme salinity. For human observers, the landscape seems endless and empty, a mirror-like expanse that emphasizes the absence of conventional ecosystems. Lake Assal in Djibouti presents another type of chemical isolation. This small lake sits in a depression below sea level and is fed by underground saline water. Its salt content is among the highest in the world, and the pH of the water is highly alkaline. Few organisms can tolerate such extreme chemical conditions. Fish and larger animals cannot survive, and plants do not grow along its shores. Only microbial life exists, forming thin mats that cling to mineral surfaces or float in shallow pools. These microbes have evolved to resist salt stress and chemical toxicity, extracting energy from mineral reactions. Despite the lake's small size compared to salt flats, its chemical extremes create a nearly lifeless environment, illustrating how composition, as well as quantity of water, limits colonization. Mono Lake in California demonstrates how natural chemical variations can produce isolation. High salinity, elevated levels of alkalinity, and high concentrations of certain minerals create conditions unsuitable for most macroscopic life. The lake does support specialized brine, shrimp, and algae, but these species are confined to specific zones, leaving large areas of the lake virtually devoid of life. Along the shores, salt deposits accumulate as water evaporates, forming crusts too harsh for plants to take root. Microbes, however, can colonize these areas, surviving in salt-rich crusts or in shallow water with high mineral concentrations. These extremophiles perform slow metabolic processes, maintaining small populations in an environment that is otherwise barren. Salinity is the primary factor that prevents most life in these regions. High concentrations of sodium chloride and other salts draw water out of cells, causing dehydration and death in plants and animals. Many organisms are unable to regulate internal water balance in such conditions. Only halophilic microbes possess adaptations that allow survival. These organisms maintain internal ionic balances, produce protective proteins, and extract energy from chemical reactions rather than sunlight. Even so, their distribution is sparse and localized. Large animals, insects, and plants are absent, highlighting the sharp contrast between microbial survival and the lack of visible life. pH and chemical composition further reinforce isolation. Highly alkaline or acidic waters disrupt enzymes, protein structures, and cellular membranes, preventing normal metabolism. Salts, minerals, and metals in the water or soil can bind nutrients or release toxic ions. As a result, areas with extreme pH are mostly devoid of life. Microbial extremophiles dominate, exploiting chemical energy sources to persist. In brine lakes, high pH and salinity often coexist, creating multiple overlapping stressors. The combination of these factors ensures that macroscopic life is excluded, leaving chemical deserts where only microorganisms endure. Satellite mapping has revealed the scale of these lifeless expanses. From orbit, the Salar de Uyuni, Lake Assal, and Mono Lake appear as vast, uniform surfaces with minimal variation in color or texture. The reflective surfaces of salt flats create mirrors that extend for kilometers, while brine lakes show subtle color gradients indicating mineral concentration. Remote sensing allows scientists to monitor changes in water levels, salinity, and chemical composition, confirming that these areas remain inhospitable for plants and animals. The mapping also illustrates how isolation is reinforced by the uniformity and extremity of these chemical environments, leaving large tracts effectively devoid of conventional life. Microbial life in salt flats and brine lakes is fascinating in its resilience. Halophiles, alkaliphiles, and other extremophiles survive in thin films of water, within salt crusts, or suspended in brine. They metabolize slowly, often using chemical reactions with minerals to generate energy. Populations are small, colonies remain microscopic, and reproduction is limited by the harsh conditions. In some lakes, microbial mats form shallow patterned layers that are visible only upon close examination. These organisms demonstrate how life can exist at the boundary of habitability. But the vast majority of these landscapes remains empty to the naked eye. Evaporation and mineral accumulation maintain the isolation of these areas. Rainwater is scarce and evaporation is rapid, concentrating salts and chemicals over time. Even where water temporarily appears, high salinity and chemical stress prevent colonization by larger organisms. Soil is minimal or non-existent, preventing plants from rooting. Small animals and insects are largely absent because they cannot tolerate the chemical environment or the lack of shelter. The result is an environment where life is limited, isolated and dominated by microbes, while larger organisms are effectively excluded. Temperature and climate influence these chemical extremes as well. Many salt flats and brine lakes exist in deserts or below sea level, where daytime heat is high and nighttime cooling is rapid. Surface evaporation enhances salinity, and winds sweep away moisture and fine sediments. The climate interacts with chemical conditions to reinforce isolation. Life that might survive in milder chemical conditions is removed by the combined stresses of temperature and dryness. The interaction of chemical, physical, and climatic factors produces landscapes where isolation is profound and enduring. Despite their harshness, these environments are of great scientific interest. Studying microbial extremophiles provides insight into adaptation, stress tolerance, and survival in conditions similar to early Earth or other planets. Experiments in situ and in the laboratory reveal how halophiles regulate water balance, repair DNA damaged by chemical stress, and perform energy metabolism in low nutrient settings. These studies confirm that microbial life can thrive in niches where macroscopic life is impossible, emphasizing both the resilience and the limits of living organisms. Chemical deserts, therefore, serve as natural laboratories for understanding the boundaries of life. In summary, salt flats, brine lakes, and chemical extremes such as the Salar de Uyuni, Lake Assal, and Mono Lake illustrate how high salinity, unusual pH, and chemical composition prevent colonization by plants and animals. Microbial extremophiles persist in small, specialized niches, while macroscopic life is absent. Satellite mapping highlights the uniformity and scale of these lifeless expanses, reinforcing their isolation. These regions demonstrate that water alone does not guarantee habitability. Even when present, chemical extremes can create landscapes that remain largely barren, emphasizing the delicate balance between environmental conditions and the ability of life to endure. In the middle of vast oceans, small islands and rocky outcrops emerge as some of the most isolated environments on the planet. Bouvet Island in the South Atlantic, Tristan da Cunha in the remote South Atlantic archipelago, and other far-flung rocks illustrate how distance, climate, and exposure combine to create lifeless zones. These islands are often surrounded by rough seas, subjected to strong winds, and lack fertile soil, making colonization difficult for plants and animals. Life that does arrive is generally sparse and transient. The isolation is physical, ecological, and environmental, leaving these rocks as testaments to the limits of life in the most distant corners of the Earth. Bouvet Island, a small ice-covered island south of the equator, is almost entirely lifeless. Its shores are steep, volcanic, and coated in ice and snow for much of the year. Strong winds sweep across the surface, and temperatures rarely rise above freezing. Soil is thin or absent, and nutrient availability is minimal. Few plants manage to establish themselves, and large animals cannot survive on the island alone. Occasional seabirds may land or nest on small patches of exposed rock, but the island provides minimal resources. Life persists in small pockets, often dependent on the sea surrounding it, rather than the land itself. Bouvet Island exemplifies how isolation, cold, and wind can prevent the establishment of terrestrial ecosystems. Tristan da Cunha, farther north in the South Atlantic, shows similar patterns on a slightly larger scale. Its volcanic slopes and cliffs are exposed to high winds and salt spray, limiting plant growth. Soils exist in some valleys, but erosion and unstable surfaces restrict their depth and fertility. Native vegetation is limited to mosses, grasses, and a few hardy shrubs. Seabirds, including gulls and petrels, use the island for breeding, providing occasional nutrients to soil through droppings. Large land animals are absent, and invasive species introduced by humans have struggled to persist in the harsh conditions. The combination of remoteness, exposure, and limited soil reinforces the isolation of the island, creating a landscape where life exists in small, fragile populations. Smaller rock outcrops around the world present even greater challenges. These tiny islands, sometimes just a few meters across, are washed by waves and subjected to constant wind and sun exposure. Soil is often entirely absent, and water is scarce or non-existent except after rainfall. Seeds that arrive are unlikely to take root, and any animals that wash ashore or fly in may struggle to find food or shelter. In some cases, seabirds may land temporarily, contributing small amounts of nutrients to the surface. Occasionally, algae or lichens form thin layers, but these are limited in size and growth. Most outcrops remain almost entirely lifeless, emphasizing the role of exposure and isolation in limiting colonization. The combination of cold, wind, and lack of soil on these islands produces a harsh microclimate. Temperatures may fluctuate daily, frost may occur, and strong winds desiccate surfaces. Salt spray from the ocean can damage plant tissues and soil chemistry. Even seeds that arrive by wind or water face extreme stress. These conditions prevent large ecosystems from forming, leaving only sparse microbial or algal communities in protected crevices. The climate itself acts as a barrier to colonization, ensuring that life remains limited to the most hardy species. Seabirds are often the first and most visible colonizers of remote islands. They can fly long distances and bring nutrients from the ocean to land through droppings. These nutrients can support mosses, lichens, and small grasses, creating microhabitats that may persist for years. However, the contribution of seabirds is limited by the size of the island and the availability of food sources nearby. Larger animals that rely on land-based ecosystems cannot survive on these islands due to the lack of continuous resources. Birds provide temporary enrichment, but they do not overcome the fundamental isolation imposed by geography and climate. Seed dispersal is a major challenge for life on isolated islands and rocks. Seeds must travel long distances across open ocean, often relying on wind, birds, or floating on water. Many seeds are destroyed by salt water, lack of moisture, or exposure to sunlight. Even when seeds arrive, they may not find suitable soil or shelter to germinate. As a result, plant colonization is slow and patchy. Only species with adaptations for long-distance dispersal or tolerance to harsh conditions can establish themselves. The difficulty of seed arrival and germination reinforces the isolation of these islands, keeping them largely barren. Marine animal settlement around these rocks is also limited. Coral or benthic communities require stable substrates, appropriate water chemistry, and shelter from strong waves. On exposed rock outcrops, wave action and strong currents prevent large communities from forming. Some invertebrates, such as barnacles or mollusks, may attach to surfaces temporarily, but populations are often small and vulnerable to environmental changes. Fish and other mobile species pass through the surrounding waters, but the rocks themselves offer limited habitat. The ocean, while full of life, does not guarantee colonization of the isolated landforms within it. Observations by scientists have confirmed the scarcity of life on these islands and rocks. Aerial surveys, satellite imaging, and field visits reveal surfaces that are largely bare, with only small patches of vegetation or animal activity. Microbial life may persist in soil or water collected in depressions, but these populations are minimal. Long-term studies show that even islands with occasional colonization by birds or drifting seeds remain largely lifeless for centuries. The extreme isolation is maintained by the combination of distance, climate, and environmental exposure. Isolation is reinforced by the size and topography of the landform. Small islands and rock outcrops lack valleys or sheltered areas where soil can accumulate and water can persist. Slopes are steep, surfaces are unstable, and erosion removes the minimal soil present. Waves and storms can wash away seeds, vegetation, or small animals. These physical constraints ensure that life cannot establish stable populations. Even if an organism arrives, the landscape may be unsuitable for survival, creating a persistent barrier to colonization. The remoteness of these islands is another limiting factor. Bouvet Island, Tristan da Cunha, and similar rocks lie thousands of kilometers from larger landmasses. The distance prevents many species from reaching them, as flying, swimming, or floating across vast stretches of open ocean is often impossible. Only highly mobile species, such as seabirds or wind-dispersed microbes, can arrive. This isolation prevents gene flow and keeps populations small and fragmented. Species that do establish themselves remain vulnerable to environmental changes, predation, and stochastic events. Temperature, wind, and exposure interact to create a harsh environment on isolated rocks. Daily fluctuations, frost, and strong gusts desiccate surfaces. Salt from ocean spray alters the chemistry of soils or temporary pools. These combined stresses prevent colonization by plants and animals that would otherwise survive in milder climates. Microbes, algae, and lichens are able to persist in sheltered niches, but larger life is excluded. The environmental combination reinforces isolation, leaving the islands barren and inhospitable. Even human attempts to study or visit these rocks highlight the difficulty of surviving there. Access is limited by rough seas, high winds, and steep shorelines. Supplies are necessary to remain on the islands, as food and water are absent. Research equipment often must be carried by boat or helicopter, limiting the duration and scope of observations. Despite these challenges, studies confirm the persistence of extreme isolation and minimal life. The islands remain largely untouched, serving as natural laboratories for understanding colonization, dispersal, and survival in remote environments. In conclusion, isolated islands and remote rock outcrops such as Bouvet Island and Tristan da Cunha demonstrate how geography, climate, and environmental exposure combine to prevent colonization by most plants and animals. Cold, wind, and lack of soil create harsh conditions, while distance prevents arrival of seeds and animals. Occasional seabird colonization provides nutrients and temporary habitat, but the majority of the landscape remains lifeless. These islands illustrate the limits of dispersal and survival, showing that even small landforms in the ocean can be among the most isolated places on Earth. In addition to natural environments, humans have created areas of extreme isolation through activity, contamination, and abandonment. Places like former nuclear test sites, the Chernobyl exclusion zone in Ukraine, and abandoned industrial complexes offer modern examples of artificial wastelands. In these locations, radiation, chemical toxicity, and physical destruction combine to create zones where life struggles to persist. Unlike natural isolation, these areas were once inhabited or fertile, but human interference has drastically altered the conditions, leaving land that is often inhospitable to most plants and animals. Over time, however, some life adapts, demonstrating resilience even in environments shaped by human impact. The Chernobyl exclusion zone is one of the most studied human-made isolated areas. Following the 1986 nuclear disaster, a large region around the reactor was evacuated and restricted. Radiation levels in some areas remain high, particularly near the reactor itself, while others have decreased over time. Soil, water, and vegetation absorbed radioactive particles, altering ecosystems and eliminating species sensitive to contamination. Human activity ceased almost entirely, leaving buildings, roads, and infrastructure to decay. In many places, life was almost entirely absent immediately after the accident. Over decades, however, scientists observed that some plants and animals returned, adapting to radiation and the absence of humans. Wolves, deer, and birds now inhabit the forests, while microbial communities in soil and water include species capable of metabolizing radioactive compounds. The zone illustrates how human-imposed isolation can both suppress life and create opportunities for extremophile adaptation. Nuclear test sites present a similar pattern. Areas in Nevada, the Marshall Islands, and Kazakhstan were subjected to explosions that contaminated soil and water with radioactive isotopes. Vegetation was destroyed, animals fled or died, and human settlements were abandoned. The resulting landscape is isolated both by toxicity and by restricted human access. Some microbial life has persisted in these areas, metabolizing radioactive compounds or surviving in soils with low nutrients and high contamination. In some cases, lichens and mosses slowly recolonize surfaces, providing a minimal foundation for other life forms. Large animals are rare, but scavengers and birds occasionally enter the area. The combination of chemical stress and lack of disturbance maintains an artificial isolation, different in origin from deserts, ice sheets, or high mountains, yet producing a similar limitation on life. Abandoned industrial areas also create isolation through chemical and physical factors. Factories, mines, and chemical plants that have been deserted often leave soils laden with toxic metals, acids, or other pollutants. Vegetation is sparse, and animals are largely absent due to both toxicity and lack of cover or food. These areas are generally smaller than nuclear sites or natural wastelands, but provide local examples of human-induced isolation. Microbial extremophiles, particularly bacteria capable of metabolizing toxic compounds or surviving with limited nutrients, may persist. Life gradually recolonizes over decades, often starting with hardy pioneer species, but the overall biodiversity remains low compared to surrounding natural habitats. Radiation, chemical contamination, and physical destruction define these human-imposed environments. Unlike natural isolation caused by climate, altitude, or geography, The stresses here are abrupt and extreme, sometimes altering the chemistry of soil and water beyond natural ranges. Radiation damages DNA, slows reproduction, and increases mutation rates. Chemical toxins disrupt cellular processes and may remain in soil for decades. Physical structures, debris, and altered landscapes remove habitat, create barriers to movement, and prevent recolonization. Together, these factors produce zones of near-total lifelessness, analogous in their barrenness to deserts or ice fields, yet distinct in their origin. Comparison with natural isolation reveals interesting contrasts. In deserts, ice sheets, and high mountains, life struggles due to environmental stresses that have existed for millennia. In human-imposed isolation, the disruption is sudden, and species may be forced to adapt quickly or perish. Microbial extremophiles in both types of environments share strategies such as slow metabolism, dormancy, and chemical resistance. But human-made zones also involve exposure to novel toxins and radiation. Recovery in these areas depends on both adaptation and the gradual decrease in stresses, illustrating that life can persist even after sudden, severe disruption, if niches remain for specialized species. Observations of recovery show that human-imposed wastelands can support surprising levels of adaptation. In Chernobyl, fungi that absorb or resist radiation grow on contaminated surfaces. Bacteria metabolize radioactive isotopes, while some plant species colonize abandoned fields and roads. In former nuclear test sites, microbial communities persist in soils previously considered sterile. Even in heavily polluted industrial zones, lichens, mosses, and certain invertebrates slowly re-establish small populations. While macroscopic diversity is limited compared to natural ecosystems, the persistence of life demonstrates resilience and the capacity of extremophiles to exploit marginal habitats. Human interference often reinforces isolation. Restricted access, contaminated soils, and abandoned infrastructure prevent migration of animals and plants into these areas. Wildlife corridors are disrupted, and species that might disperse naturally cannot establish populations. In some cases, abandoned areas remain isolated for decades, preventing recolonization and maintaining lifeless expanses. Unlike natural isolation, which is often permanent or predictable, human-imposed isolation can be partially reversed if cleanup or restoration occurs. However, in the absence of intervention, these zones act as artificial deserts, chemically and physically inhospitable to most life. Even when human-imposed isolation diminishes over time, environmental stresses remain significant. Radiation decays slowly, toxins persist in soil and water, and physical structures may continue to prevent soil formation or vegetation growth. Microbial extremophiles may remain dominant for centuries, while macroscopic plants and animals remain rare or absent. This contrasts with natural isolated areas, where life may slowly adapt to environmental extremes over millennia. Human-created wastelands provide a unique opportunity to study the limits of adaptation under sudden, severe stress. In summary, nuclear test sites, the Chernobyl exclusion zone, and abandoned industrial areas represent examples of human-imposed isolation. Radiation, chemical toxicity, and physical disruption limit colonization by plants and animals. Life that persists often consists of microbial extremophiles or hardy pioneer species, slowly adapting to novel stresses. Comparison with natural isolation highlights differences in origin, speed of environmental change, and recovery processes. Observations confirm that even under human-induced extreme conditions, life can endure, though the landscape remains sparsely inhabited and profoundly isolated. illustrating the capacity of organisms to persist in artificial wastelands. The study of Earth's most isolated places is not only a record of where life struggles to exist, but also a window into the limits of life itself. Deserts, high mountains, deep ocean trenches, volcanic wastelands, salt flats, remote islands, permafrost, and human-made exclusion zones all show the ways that environmental extremes shape ecosystems and restrict survival. Scientists examine these regions to understand how organisms cope with harsh conditions and what adaptations allow life to endure where most species cannot. The insights gained inform fields as diverse as ecology, climate science, and astrobiology, providing lessons that reach beyond our own planet. Ecologists study isolated zones to understand how environmental stress controls species distribution. These areas demonstrate that life is not evenly spread across the planet. Instead, the presence or absence of species depends on temperature, water, chemical composition, radiation, altitude, and isolation. By observing which organisms survive, scientists can predict how ecosystems respond to extreme events or changing climates. Microbes in salt flats, extremophiles in volcanic soils, and high-altitude plants offer living examples of resilience. They highlight that the boundaries of life are determined not only by resource availability, but also by the interplay of multiple environmental stresses. Extremophiles are central to understanding these limits. Microorganisms that thrive in high radiation, intense salinity, extreme temperatures, or chemical toxicity reveal the cellular and biochemical mechanisms that support survival. They can repair DNA damaged by radiation, extract energy from chemical reactions in the absence of sunlight, and regulate water and ion balance under osmotic stress. Studying these adaptations allows scientists to see how life can persist under conditions once thought uninhabitable. These findings also demonstrate that life does not require moderate or familiar environments to survive. Instead, it can adapt in ways that challenge our traditional definitions of habitability. Insights from extremophiles extend into astrobiology, the study of life beyond Earth. Isolated and extreme environments on our planet are analogues for other worlds. For example, the icy surface of Europa, a moon of Jupiter, may host subsurface oceans with high pressure and low temperatures, similar in some ways to subglacial lakes or deep ocean trenches on Earth. The dry, salty soils of the Atacama Desert resemble Martian regolith, where water is scarce and chemical stress is high. Observations of extremophiles inform the types of life that might exist in these conditions and guide the design of experiments and missions searching for extraterrestrial organisms. Understanding how life survives on Earth's extremes provides the blueprint for seeking life on Mars, icy moons, or even distant exoplanets. Climate science also benefits from the study of isolated regions. Polar ice sheets, permafrost, and high-altitude plateaus contain records of temperature, precipitation, and chemical composition over thousands or millions of years. Microbial life within these environments shows how organisms respond to slow or sudden environmental changes. Studying the survival strategies of extremophiles under freezing, drought, or chemical stress informs models of resilience and adaptation under global climate shifts. By observing how isolated ecosystems cope with stress, scientists can better understand the impact of climate change on more populated and biodiverse regions. Artificially isolated areas provide additional insights. Human-imposed wastelands, including abandoned industrial sites or nuclear zones, reveal how ecosystems respond to abrupt, severe disturbance. Extremophiles in these zones show that life can persist even when chemical toxicity or radiation would prevent conventional organisms from surviving. Comparing recovery in human-imposed isolation with natural isolation highlights the mechanisms through which organisms recolonize, adapt, and establish ecosystems under extreme pressure. This knowledge informs environmental remediation, conservation strategies, and risk assessment for contaminated regions. Studying isolated places also enriches our understanding of evolutionary processes. In regions where life is scarce and populations are small, selective pressures are intense. Organisms must survive with limited resources, extreme temperatures, or chemical stress. Mutations that provide even slight advantages are rapidly reinforced, leading to highly specialized adaptations. Extremophiles demonstrate how evolutionary pathways can diverge from familiar patterns observed in temperate ecosystems. Isolated environments act as natural laboratories for observing the interplay between environmental pressure and biological innovation. The philosophical significance of these isolated zones is also profound. Observing landscapes devoid of conventional life prompts reflection on the resilience of living organisms and the fragile balance that allows life to exist. Life persists in niches that seem impossible. Microbes under thick ice, mosses on windswept cliffs, extremophiles in toxic soils. Each of these organisms is a testament to the power of adaptation and the capacity of life to exploit even the harshest conditions. The extremes of Earth remind us that habitability is relative and that life can endure in forms and places far from human expectation. Human perception of isolation is challenged by these environments. What appears lifeless is often teeming with microscopic activity, unseen by the naked eye. The subtle signs of survival, biofilms on rocks, microbial mats in brine, tiny plants in volcanic cracks, demonstrate that life is persistent even where conditions are extreme. Studying these areas fosters appreciation for the invisible resilience that sustains ecosystems and shows that apparent emptiness does not equal true sterility. Instead, isolation magnifies the ingenuity of life, revealing strategies that are otherwise hidden in more hospitable landscapes. Finally, examining isolated zones allows scientists to anticipate potential threats to biodiversity. Understanding the conditions that limit colonization, the adaptations that permit survival, and the processes that drive recovery provides insights into conservation priorities. Some isolated regions, whether natural or human-made, act as refuges for unique species or as testbeds for understanding ecosystem responses. Knowledge gained in these extreme areas informs how we manage environments under stress, whether from climate change, human activity, or natural hazards. In conclusion, studying Earth's most isolated places matters for multiple scientific fields. They illuminate the boundaries of life, the adaptations of extremophiles, and the mechanisms of survival in extreme environments. Insights gained inform astrobiology, guiding the search for life beyond Earth, and climate science, helping predict responses to environmental change. These regions also offer philosophical reflection on the resilience of life and the ingenuity of adaptation. Across deserts, ice sheets, high mountains, deep seas, volcanic wastelands, chemical flats, remote islands, and human-made zones, isolation shapes life, challenges survival, and reveals the extraordinary persistence of organisms in conditions that seem inhospitable. They remind us that while life is rare in some places, it never disappears entirely, always finding ways to endure.